Hello and welcome. My name is Mac Hopkins and I am a Technical Product Manager at BD Biosciences. Thank you for joining me for my presentation entitled BD Rhapsody Single-Cell Analysis System, a robust microwell-based single-cell partitioning system for high-dimensional biology research. During today's webinar, we will cover the underlying technology of the Rhapsody analysis system and the currently available assays, including our newly released full-length BDJ kit. We will also review the work BD Biosciences has been doing with Immudex, and finally, discuss a multiomics application utilizing dextramer technology. Let us begin our journey by understanding what the BD Rhapsody Single-Cell Analysis System is. The Rhapsody system is composed of two instruments. The first is the Express, which acts as the workhorse of the two. This small, lightweight device is easily portable, requires no electrical input, and is where all the wet lab steps take place, such as cell capture and partitioning. Diving further into the specifics of how this instrument works, the Express is comprised of two sliders located on the front and left-hand sides. The front slider controls the position of two tubes, one for collecting waste and one for collecting the cell capture beads after lysis. The left-hand slider controls two magnets which allow for the manipulation of the cell capture beads at various points of the assay. I will explain more about this in the upcoming slides. On the Express sits a disposable cartridge that has over 200,000 microwells used to capture individual cells and cell capture beads. There is an inflow port on the top where reagents are added, and an outflow port underneath that directs liquid to the waste container or collection tube. The design of the cartridge ensures minimal chance of clogging, leading to wasted reagents and sample loss. The reagents are introduced to the cartridge using one of the two electronic Gilson pipettes, which come with the system. These pipettes are specifically calibrated for optimized dispensing speed of reagents through the cartridge, so cells and beads are not disturbed nor washed away. Imagine you have added your cells to the cartridge and are now pipetting in the cell capture beads. If the dispensing speed is too fast, all the cells could be lost at the second step of the assay. This is too difficult to control manually, but the electronic pipettes take the work and the worry out of the picture. The last piece of equipment is the Rhapsody Scanner. This optional instrument serves as both a hemocytometer for cell counting and viability assessment, as well as a real-time QC instrument reporting metrics of your experiment as you are performing it. The scanner is both a fluorescent and light microscope capable of counting the number of cells captured and the doublet rate, among other metrics. This information allows you to make informed decisions while you are conducting your experiment and be in total control of how to proceed. Now that we have covered how the instruments work, let's see them in action. This cartoon represents a cross-section of the cartridge with the partitions on the bottom representing the microwells. After first preparing the cartridge by washing it with buffer, the residual liquid is removed and your single-cell suspension is added. The cells are allowed to settle, and then the cartridge is imaged by the scanner to count the number of cells captured. The cells have been stained with calcium AM and DRAC7, a live and dead stain respectively, so the scanner can discriminate between live and dead cells based on their fluorescence. This is a representational image of what the scanner is seeing, and you will notice both green live cells and non-fluorescing debris are present. The live cells will be counted, and this number, as well as the multiplet rate, will be reported back to you. At this point, you can make a critical decision as to if the experiment is worth proceeding with. Say you aim to capture 10,000 cells, but the scanner reports only 3,000 are present with a multiplet rate of 10%, which is 7% higher than expected. These values indicate something has gone wrong, and it is likely the cells are sticking to each other. The experiment can be stopped here, and the cell prep re-optimized to address this issue. However, if the number of cells captured and multiplet rate are within your expectations, you can proceed forward. Now that the cells are captured, the next step is to add the magnetic cell capture beads. These are loaded to excess to fill every well with a bead, even if it does not have a cell. Wells with only a bead will not produce any data. Once the beads have settled, the scanner is used to assess bead uniformity in the cartridge. Receiving a pass, we then wash the cartridge twice to remove any excess beads. The cartridge is imaged again to assess the number of wells that have both a cell and a bead. This is an important metric because only wells with both a bead and a cell will produce data. This value is the theoretical upper limit for the number of cells in the final dataset and will be used to calculate the total amount of sequencing needed. The cell retention rate is also calculated to ensure that most of the cells remained in their wells during bead loading and washing. If a large number of cells were lost during these steps, the experiment can be stopped and further sample processing can be done. The orientation of the well now has the cell at the bottom and the bead on the top acting as a plug. The beads almost entirely fill the well, so only one bead can be in a well. The lower magnet is then engaged to bring the bead to the bottom of the well, bringing it into closer proximity to the cell and keeping it in place during lysis. Lysis buffer is then added to the cartridge to release the cellular content, and a short incubation allows for binding to the cell capture beads. After 2 minutes, the upper magnet is engaged to lift the beads out of the wells, and buffer is flowed through the cartridge to remove the beads and collect them in a separate tube for further processing. The cartridge is scanned one more time to confirm greater than 90% of the beads have been recovered. The beads are then washed to remove the lysis buffer before cDNA synthesis. On this slide, you will see a representative image of the cell capture bead and one of the DNA bead oligos. The bead oligo includes a few critical sequences. The first is a poly-T sequence that captures anything with a poly-A tail, such as mature mRNA or oligo-conjugated antibodies. I will come back to these antibodies later in the webinar. The next sequence is a UMI, or unique molecular index, which allows for molecular counting and accurate assessment of gene or protein expression. The sequence of the UMI is different for every bead oligo on the same bead. Continuing to the left, there is the cell label sequence that acts as a molecular barcode and lets us trace the RNA and protein information back to specific cells. Finally, the sequence closest to the bead is a universal oligo primer binding site used for PCR amplification. During lysis, anything with a poly-A tail will hybridize to the poly-T on the bead. Again, this will include both mature mRNA and oligo-conjugated antibodies. A copy of what is bound is made through cDNA synthesis, and an EXO-1 treatment degrades any unoccupied bead oligos, reducing noise in the dataset. The cDNA is covalently bound to the beads and can be stored for up to 4 months at 4 degrees Celsius. These beads are then used as the input for library preparation. One of the major concerns when performing single-cell experiments is how many cells are initially captured and how many of those cells are found in the sequencing data. A major hallmark of the Rhapsody system is its high cell capture rate and retention through sequencing. This table shows the results of 75 experiments using GERCAT and Ramos cell lines. 5,000 cells were loaded into 75 different cartridges, done in replicates of 2 to 5 per experiment. The live cell recovery rate for these experiments was nearly 75%, with an overall variability of about 10% and a replicate variability of 3.4%. This high capture and retention rate means you will have richer data sets and more cells to analyze, which is particularly important for precious samples. Now that we have stable cDNA, let us talk about the available downstream library preparation options. There are two main mRNA assays for the interrogation of gene expression: the whole transcriptome approach and the targeted approach. Both can process cells and nuclei. However, the whole transcriptome approach is generally preferred for nuclei. Each method has its pros and cons, and careful selection during your experimental design will help produce the most worthwhile information while balancing the cost of sequencing. Whole transcriptome analysis, or WTA, is more appropriate when in discovery mode. This assay uses all the cDNA as templates, meaning all transcripts are represented in the final dataset. Since this assay is unbiased, isoforms and variants will also be included. This approach allows you to characterize the total transcriptional profile of a cell, which brings the most insight when studying novel biological systems or unknown responses to a stimulus. Due to the richness of WTA datasets, the sequencing requirements are greater than the targeted method because constitutively expressed genes like housekeeping and mitochondrial ones make up a large portion of the data. Achieving a high enough sequencing depth for the genes of interest can be challenging, leading to rare transcripts or lowly expressed genes missing from the dataset. On the other hand, the targeted mRNA assay uses a priori knowledge, either public datasets or WTA data, to design gene-specific primers to amplify only a small subset of genes. The Rhapsody targeted mRNA library prep utilizes a nested PCR approach for greater specificity of the targets of interest. Since gene-specific primers are used, a high confidence reference is needed for this method to be effective, and is most appropriate for well-characterized systems like PBMCs or CAR T cells. Unknown isoforms or variants can be missing from the final data set because primers were not included. However, better resolution of rare or lowly transcribed genes is possible due to the PCR-based method. The number of genes included in this assay usually ranges from 100 to 500, leading to the significantly lower sequencing costs than the WTA approach. Once the appropriate mRNA method is chosen, additional assays can be included to characterize other aspects of your cells in a truly multi-omic approach. If working with PBMCs or purified T and B cells, a VDJ component can be included in your experiment to interrogate the immune repertoire. We have recently launched our Tcr Bcr Multimic Assay for human samples, with a mouse option coming later this year. This kit includes everything needed to profile the full-length T and B cell receptors and is compatible with either WTA or targeted mRNA assays. Additionally, the Tcr Bcr Multimic Assay can be used with our oligo-conjugated antibodies, allowing for cell surface protein characterization and sample multiplexing. Our pipeline has been updated to handle these data and takes all the work out of your hands. The full-length amino acid sequence, shown on the slide, of the Tcr or Bcr, as well as myriad other information, is reported back once the pipeline finishes. This easy-to-use assay seamlessly integrates into your current Rhapsody experiments, bringing a fuller understanding of the underlying biology of your samples. Moving on from RNA-based assays, I will now switch to our antibody offerings. The first oligo-conjugated antibody, or ab-oligo technology, I will discuss is our sample multiplexing kit. The sample tag antibodies In the sample multiplexing kit are 12 tubes of the same antibody specificity that have a slightly different DNA oligo bound to them. This oligo has a poly-A tail for hybridization to the cell capture bead, a primer binding site for amplification, and most importantly, a unique barcode sequence. These barcoded antibodies allow you to stain 12 different single-cell suspensions and mix them together for processing in the same cartridge. Higher number of cells and samples can be processed this way, making these experiments even more cost-effective. This technology is available for both human and mouse samples, but custom options are possible. Finally, since each cell will be tagged with a specific barcode, discrimination and removal of doublets from the dataset is much easier. Let me show you an example of this. In this experiment, purified mouse T and B cells were split into 6 aliquots each and stained with individual sample tag antibodies, utilizing the entire kit. The 12 aliquots were then mixed in equal ratios and loaded into the same cartridge. After sequencing and data analysis, you can see 3 distinct populations on this Tisney plot. The top represents T cells, the bottom B cells, and the smaller one to the left-hand side are multiplets of B and T cells. This is a very obvious example, and even without the sample tag antibodies, we can identify this group as being unexpected. Multiplets make up almost 12% of these data, and a lot of them are located in this group. However, there are multiplets of T and T cells, as well as B and B cells, which would not show up here. This represents a more realistic scenario when it is not so simple to identify which cells are actually multiplets. Using the sample multiplexing antibodies, even these hidden multiplets can be excluded from the dataset as long as they came from a different sample, having different sample tags. Removing all multiplets, these data can be remapped, and now we can see 2 distinct and clean populations of cells. The second AB oligo technology I will discuss is called AB-seq. AB-seq, or antibody sequencing, is an antibody-based method for interrogating cell surface proteins in conjunction with a single-cell transcriptomics assay. On this slide, you can see the structure of our DNA oligo-conjugated antibodies. At the very terminal 3' end of the oligo tag is a poly-A tail that hybridizes to the cell capture bead during lysis. Moving towards the 5' end, the next sequence is the antibody-specific barcode. The base pair sequence of this barcode is unique for each antibody specificity and allows for the identification of which antibodies bound to which cells. Further to the left is the unique molecular index, or a UMI, which is important for the elimination of PCR or sequencing artifacts from the dataset. Additionally, the UMI allows for a quantitative assessment of how many antibodies bound to each cell. Finally, the most 5' sequence is a PCR handle used for amplification and library preparation. The AB-seq portfolio currently offers over 400 oligo-conjugated antibodies split almost evenly between human and mouse specificities. Every AB-seq antibody is rigorously tested and compared to its fluorescently labeled twin to assess specificity and sensitivity. Those that pass our internal controls are titrated and sold off the shelf in 25 test size aliquots. Finally, each AB-seq antibody has a shelf life of 2 years. I am often asked, what is the largest number of antibodies that can be used in one run? To answer this question, we decided to push the limits of our technology. In this experiment, Jurkat, Ramos, and THP1 cell lines were mixed together and stained with a core 20-plex of AB-seq antibodies. Each of these cell lines represents a different major subset of cells in peripheral blood, T cells, B cells, and monocytes respectively. This core 20-plex of antibodies was designed such that a signal for each marker would be seen for each of these cell types, but at varying intensity. To this core panel, 20 more antibodies were added in sets of 20 until a total of 100 AbSeq were simultaneously stained. Each level of plexi, 40, 60, 80, and 100, was assessed separately to determine if an increase in the number of antibodies pooled impacted the performance of the core 20. At the 100-plex level, certain markers were chosen which are specific for only one subset of cells in the mixture. With the expectation of dropouts, this allowed for the evaluation of specificity at such a high plexi. The stained cell mixtures were then processed through the Rhapsody Single-Cell System, libraries were prepared, and then sequenced. On the next slide, I will briefly walk through the metrics of the experiment before showing the staining results. This first chart represents the median AbSeq targets, which you can think of as specificities, identified per cell at each plexi. From 20 to 80 plex, each antibody is seen on every cell, but at 100 plex, the median antibodies identified is less than 100, which, as I mentioned in the previous slide, is expected based on antibody choice. Additionally, the antibodies included for each successive 20-plex were chosen to include high, medium, and low expressors to showcase the sensitivity capable of this technology. Understandably, since the number of antibodies increased with each successive staining, higher amounts of sequencing data were required to maintain similar sequencing saturation for each plexi. This graph simply displays each plexi had approximately the same sequencing saturation. Now, let's move on to the actual results. This heat map has a lot going on, but let me walk you through it step by step. The first 20 lines on the y-axis are the core 20-plex. Each successive 20 lines are the next batch of antibodies supplemented to the core 20, with the final lines being the 80th to 100th antibodies. The labeling on the x-axis is broken out by the AbSeq plexi and the cell line. For example, the first column is Jurkat cells with only the core 20 antibodies stained. The next is again Jurkat with 40 antibodies, etc. Focusing only on the core 20 plex, you will notice that as you increase the number of antibodies total, the relative expression of these 20 is unimpacted, and the expression remains stable up to 100 total antibodies across all three cell lines. One will also see that each cell line has variable expression of this 20-plex, showing that not only can highly expressed proteins be assayed at high plexi, but lowly expressed proteins are not lost in the fray. Moving down the heat map, a similar pattern emerges where the successive addition of more antibodies does not impact the resolution of the previous plexi. Even at the higher plexi samples, here I have boxed 60 and 80 antibodies. The expression is unchanged when moving into even higher numbers of antibodies. This simple experiment, I hope, has shown, regardless of the plexi required for understanding your biology, that AbSeq is scalable, sensitive, and consistent. Oftentimes, experiments are stalled once sequencing is completed. But with the BD Rhapsody system, we've made processing your data as simple as possible. FASTQ files, the raw data from sequencing runs, are processed through our primary analysis pipeline. The pipeline can be run either locally or on a user-friendly cloud-based platform called 7 Bridges Genomics. This is an example of the user interface when starting a pipeline run on 7 Bridges. On the left-hand side, you can upload the FASTQ files, an AbSeq reference file, if included in the experiment, and the genome references. You can additionally modify the pipeline settings and specify sample names if sample tags were included. Once finished, hit Run, and the pipeline will process the data set using 7Bridges resources. Utilizing this option is a nice alternative to complicated pipeline runs requiring coding and local servers. It is also easy to share these runs with colleagues or collaborators so everyone is aligned and the data is readily accessible. When the pipeline finishes, you will receive an email letting you know your data is ready. Logging back in, you will see the pipeline run completed and have the option to download multiple files produced during the run. These include, but are not limited to, overall metrics reports and gene by cell matrices used for secondary analysis. Any cost incurred by the pipeline is covered by BD. The final step in the workflow is data exploration. BD continues to support you here with the usage of SeatGeek. This user-friendly secondary analysis tool is similar to FlowJo, if you're familiar with it, and aids you to quickly find meaningful insights in your data. No coding is required for the easy creation of publication-worthy figures. Having now covered our complete portfolio, let's move on to one of our ongoing collaborations. In this section, I will briefly cover an additional multi-omic layer available through the use of dextramers produced by Immudex. This cartoon represents an Immudex dextramer. The flexible dextrin backbone has multiple MHC peptide complexes conjugated for binding to T-cell receptors. Additionally, multiple fluorophores are present, allowing for fluorescence-activated cell sorting of dextramer-positive cells. Comparing cells stained with dextramers and tetramers, you will notice that the former can detect both low and high affinity binding events, whereas the latter is only able to discriminate high affinity binding. Both negative controls show some background, but nothing unreasonable. Using this base technology, it was adapted to be compatible with the Rhapsody system. In this cartoon, you will notice all the same components are still present, but a new DNA oligo tag is added to the dextrin backbone, allowing for the dextramer to hybridize to the Rhapsody cell capture beads. This oligo tag also has barcode information, similar to AbSeq and sample tag antibodies, that identifies what dextramer bound to which cell. The PE fluorochrome gives you the option to enrich dextramer-positive cells prior to the Rhapsody workflow. Integrating dextramers into a single-cell experiment is very simple. Once the peptide pool is designed, a single-cell suspension is stained with dextramers and AbSeq antibodies if being used. The cells are washed and can then be sorted for dextramer-positive enrichment. The standard Rhapsody workflow is then followed for cell capture, cDNA synthesis, and library preparation. After sequencing, our pipeline identifies dextramer reads with the inclusion of a supplemental reference and reports the read counts in the gene by cell matrix. Visualization and deeper analysis are accomplished in SeatGeek with no special plugins or modifications needed. With the background of dextramers covered, let us look at a real-world example of how they can be used on the Rhapsody Single-Cell Analysis System. In this experiment, PBMCs were stained with a library of 13 dextramers, 8 positives and 5 negatives. The positive dextramers included peptides specific for flu, cytomegalovirus, which infects more than 50% of adults worldwide, and Epstein-Barr virus, which infects nearly everyone. A CD8 flow antibody and live dead stains were used to sort live cells positive for both CD8 and dextramers. Additionally, a 16-plex AbSeq panel was used that included CD8, CD8 positive cell activation markers, and other lineage markers to assess the sort purity. Finally, a panel of nearly 400 genes was used for the assessment of gene expression. After staining, 5,000 cells were sorted on the Melody that were double positive for CD8 and dextramer. 3,000 of these cells were captured on the Rhapsody and sequenced on Illumina NovaSeq 6000. The FASTQ files were run through the Rhapsody analysis pipeline and analyzed in SeqGeek. These plots show the initial quality control of the cells. In the first graph, knee calling is used to remove potentially dead cells, which made up about 1.6% of these data. Furthermore, plotting CD14 versus CD19 ABSeq signals allowed for gating of macrophages and B cells from CD8 positive T cells. This showed that our sort was not completely pure. Finally, plotting a dextramer negative control against the CD8 ABSeq signal was used to set a threshold for dextramer positive cells. Now that the data is cleaned up, assessment of individual dextramers could be accomplished in one of two ways. This method of gating is very similar to how we excluded CD14 and CD19 positive cells previously. Plotting CD8 against each individual dextramer, double positive populations can easily be identified and noted. Additionally, the total percentage of those double positive cells is reported. Interestingly, for one of the CMV dextramers, a mid and high signal could be determined and shown in the bottom left-hand corner. Alternatively, this method of gating utilizes the signal intensity or read count for each dextramer to identify a positive signal. The large plot on the left-hand side has the combined intensity of the negative controls where the background threshold can be established. Using this value, the 8 positive dextramers can be gated for their positive signal, including the one, CMV, having both a mid and high signal. Either method is acceptable and should result in similar double positive percentages. The cleaned up and gated data were then dimensionally reduced based on both gene and protein expression using PCA and t-SNE, then plotted in two-dimensional space. You can see distinct islands are formed in this t-SNE plot reflecting the global structure of these data. Overlaying the dextramers, CD14 and CD19 signals, localization of specificities is evident with some islands having only one dextramer or antibody signal. As an additional QC step, the CD18 ABSeq signal was assessed resulting in 2 clusters with little to no signal, these being the CD14 and CD19 positive populations previously identified. Individual dextramers can be overlaid on the t-SNE plot to gain a better understanding of their overall expression. These 5 plots represent the negative controls. The first plot is a non-specific marker, so its presence in all islands is not surprising. The remaining 4 negative controls show little to no signal in the CD8 positive islands and have primary localization to CD14 and CD19 positive cells. There is always an expectation of some background noise, but the gating previously done helped determine what is real signal versus noise. Moving to the positive controls, we see the first 3 dextramers made up 1% or less of the total population. So identifying cells positive for them is difficult, though some signal can be seen. The remaining dextramers have a robust signal, with most localizing to individual islands, meaning those cells are specific for that dextramer and also have unique gene and protein expression profiles when compared to the other cells. There is only one overlapping island with co-localization of Epstein-Barr virus and cytomegalovirus dextramers, though the cytomegalovirus signal appears to be stronger. Further investigation is needed to understand the presence of both dextramers here. Finally, the frequency of dextramer-positive cells was compared between flow cytometry and the Rhapsody data. Even though only 3,000 cells were captured in the single-cell dataset, the dextramer frequencies are comparable to the flow results. This shows that little bias is introduced during the Rhapsody workflow, preserving the cellular ratios of your sample. With that, I'd like to thank you for joining me for my presentation on the BD Rhapsody Single-Cell Analysis System. If you are interested in learning more, please find additional information on our website or reach out to your local sales representative. I hope you have a lovely day.